everybody. Today we are going to be learning about DNA replication, but before we get into that, I would like to review a couple of things from you that you might have learned a little bit about during some of the previous units. First thing we're going to review is the structure of nucleic acids. So nucleic acids include DNA and RNA. We're going to focus on DNA. There are two main parts. There are the base pairs, which you can see in orange in these pictures, and the backbone, which is in green and has a sugar group, or a sugar, and a phosphate group. The formation of DNA comes out to be a double helix because of the bonds between the base pairs. As you'll be able to learn about a little later, these base pairs act like a zipper for the DNA molecule. They hold it together when necessary, but it can come apart with the right pieces. So why does replication occur? It occurs because when we are making new cells in our body, every cell needs an identical full copy of DNA. And remember, every cell has 23 pairs of chromosomes or 46 individual ones. So you start with a cell that has 46. It replicates here where the purple arrow is. So that same cell will have 92, then it'll split into 46 again. And then each of those cells, when it needs to make new ones, will replicate, have 92, and split. This process continues over and over and over again as long as our body needs to continue to make new cells. So, for replication, there's a few vocab terms that you guys will need to know. First is called the replication fork, which is a Y-shaped area, as you can see right there, that forms when the DNA helix unwinds. The DNA helix unwinds with the help of DNA helicase, which is an enzyme that helps unwind the DNA double helix during replication. Throughout this set of notes and in some of your packets, you will notice this as a triangle in some of the pictures. I also remember that the helicase unwinds the double helix because helix sounds like the first half of helicase. DNA polymerase is an enzyme that catalyzes, which means to help build, the new DNA molecule. This will be seen as a rectangle throughout this presentation and your packets. The last one is DNA ligase, which is an enzyme that takes separate pieces of DNA and puts them back together. This picture here shows these separate pieces of DNA. This piece was also separate from there, and this little green circle is the helicase. It helps to put these pieces back into one big strand. Now we're going to get into the steps of replication. So the first one is unwinding and separating DNA strands with DNA helicase. So here's a full DNA strand that has been unpackaged from its chromosome. The body will add helicase, which again I said earlier is a green triangle, which acts like a zipper pull to unzip the helix or separate the bonds in the DNA molecule. The second step of replication is making a new molecule. So a new molecule or strand of DNA is formed when DNA polymerase adds bases to the pairs. A always goes with T when it comes to DNA, and C always goes with G. So as you can see down here in this picture, I have some C's, T's, G's, and A's all drawn out. So up here it goes TC, AG, TC, AG. Down here it's AG, TC, AG, TC. The helicase has already unzipped this molecule. And these rectangles are going to be the polymerase for each half of the molecule, because each strand is going to get a new copy to go with it. This bottom strand is called the leading strand. Leading strand. Because as I drew in this arrow, it's going towards this replication fork Y, or towards where the helicase is separating the molecule. This is usually one big long piece. The one on the top here is going away from that fork. So it is called the lagging strand. Remember, leading goes towards the fork, lagging goes away from the fork. The lagging strand usually also comes out in pieces. So there's two separate pieces here, and as I said earlier, there's another enzyme called DNA ligase, which is this little circle here, that will help put these two pieces together. The other thing that's really cool about DNA polymerase and important to remember is, let's say this polymerase made a mistake and put an A in here. So it made a mistake and put an A because a C is always supposed to go with a G, and that's a G. The DNA polymerase can then proofread this, get rid of this one, and put the correct one in. It's called proofreading, just like when you're writing a paper. So the polymerase can proofread and make sure it actually matches them up right. This is important because it helps prevent mutations or genetic abnormalities. And finally, once the polymerase, the ligase, and the helicase have all finished their jobs and separated the original molecule completely and copied all of the base pairs, it ends up making two different strands of DNA, or two different molecules. So you have two DNA molecules that are formed from one original. And this is going to happen for every chromosome in the cell. So all 46 will get duplicated. Remember, one chromosome is one DNA molecule.